Dr. Yehuda Arieli will take us for a quick dive into the brain and will speak about the natural protection against CNS-oxygen toxicity.
Okay, the combat divers and recreational divers commonly use rebreathers in which oxygen-enriched gas mixture enable divers to make a longer dive more comfortably with no bubbles to cause noise or other disturbances. These benefits, however, are frequently accompanied by a significant increase in the risk of CNS-oxygen toxicity and its typical common symptom, grand mal convulsions. High levels of reactive oxygen species and nitrogen species are a major factor in the generation of CNS-oxygen toxicity. Under normal physiological conditions, there is equilibrium between the generation of RS and RNS and their neutralization by intrinsic antioxidants. However, in stress situations, such as hyperoxia, this balance is upset by increased production of RS or RNS. This might lead to oxidative stress and cell damage. We have a lot of demonstration of the fate of ROS and RNS. I'll do it quickly. As we breathe oxygen, we produce superoxide, which is one of the vicious member of the ROS family. Superoxide with NO produce peroxynitrate, which is another member of the RNS family. And on the other route, we have a reaction with superoxide dismutase to produce oxygen and hydrogen peroxide. And as you can see, this reaction is more than 3 times faster than this reaction. But we will follow this reaction to see the fate of ROS. With the enzyme glutathione peroxidase, we neutralize the hydrogen peroxide to create water. And this reaction is executed with the assistance of glutathione reductase and G6PD. But on the way, we produce more superoxide. And that raises the question, what is the balance between the neutralizing of ROS and creating ROS? The purpose of this review is to integrate the data collected in our laboratory over the past decade in order to provide a clear picture of the mechanism involved in CNS-oxygen toxicity. We follow 2 separate lines of investigation. Heat acclimation is known to provide cross-tolerance to various forms of stress and in different organs. Suspected players in this game are ROS scavengers, the heat shock proteins 70 and 90 family, and other proteins involved in energy management and cell integrity and levels of mitochondria membrane potential. The second route, there is evidence that preconditioning to high levels of oxygen under normal baric conditions provide protection against hyperoxic lung injury. So we follow these 2 routes in order to understand the mechanism. The general methods, you can see here an example of the fragment of EEG recording from the rat at a pressure of 608 kilopascal in air in this fraction, and in oxygen after 20 minutes of getting used to the chamber and to the conditions. And as you can see here, the change in the recording, we get more and more spikes, higher spikes, and after about 2 seconds or something like 10 seconds, we can see the convulsions appears. Heat acclimation was gained by exposing the rat to 32 degrees for 4 weeks. EEG, as you saw, was recorded continuously until the appearance of the first electrical discharge. Immediately thereafter, rats were sacrificed and samples were taken from the hippocampus and frontal cortex for biochemical investigation. Brain samples were taken from the heat-acclimated rat after HBO exposure for measurement of heat shock protein 72, copper zinc superoxide dismutase, manganese sod, glutathione peroxidase, and catalase. Level of antioxidant enzymes and HSP72 were measured using Western blot, two-dimensional electrophoresis, and protein identification, which were performed after HBO exposure. The first slide of the graphs here show you the level of the HSP and the connection to the latency. As you can see here, the latency after 4 weeks of acclimation was very high and dramatically high compared, and then following 4 weeks of deacclimation, the HSP level was declined, and you can see that we have a very good linear relation between the latency and the level of HSP72. The level of copper zinc sod also increased following heat acclimation, and that was gained by Western blot. And when we investigated the fate of some other proteins, you can see that in two-dimensional electrophoresis, you can see changes in all these proteins. Alpha-synuclein also is known as heat shock proteins, was past probably phosphorylation, also the tyrosine 3 monooxygenase. And what is more interesting is the changes in the mitochondrial ATP synthase, alpha subunit. We have this subunit is embedded in the inner mitochondrial membrane, and you can see it's also as a proton pump and producing ATP with this very complex. We found the alpha unit increased dramatically following this, following the heat acclimation. No. Okay, sorry. The third branch of this investigation was mitochondrial functioning. Mitochondria are both the major source of ROS production and the major target for its destructive effects. Mitochondrial inner membrane potentials was investigated using the dye JC1. Samples from the hippocampus and frontal cortex were investigated under normal conditions and oxidative stress by giving 18 micromolar of hydrogen peroxide. Here's a sample of the diagram that we gained by with the JC1 dye. You can see here the mitochondria suspension from, let's say, normal hippocampus with no stress, and the same mitochondria suspension after giving the hydrogen peroxide, and we could compare or get the ratio between the slopes and the gap between the membrane potentials when we know that there is a linear correlation between the dye and the membrane potential. So what we have is the slope ratio when we compare the normal mitochondria with those who are under oxidative stress. in the cortex of the non-acclimated rat and the cortex of heat-acclimated rats, and you can see no changes. We have a ratio of 1, meaning that there is no change in the slopes. On the other hand, in the hippocampus of the non-acclimated rat, we have a significant change compared with the hippocampus of the heat-acclimated rat, which is very much the same like in the cortex. The delta peak, when we calculate the normal minus the H2O2, and we have, again, cortex of the frontal cortex of the control rats versus the cortex of heat-acclimated rat, there is no significant changes. And again, significant changes was gained in the hippocampus when we compare the hippocampus of the control rat compared with the heat-acclimated rats. To summarize the heat acclimation study, heat acclimation prolongs latency to CNS oxygen toxicity. It increases the level of heat shock protein 72, copper, zinc, sod, and number of other enzymes, adenylat kinase, ATP synthase. It also induces a modification of certain proteins, such as alpha-synuclein, that may contribute to energy management and integrity of the cell. Levels of other antioxidants, enzymes, were not significantly affected in this study. Heat acclimation reduces the inner mitochondrial membrane potential in the hippocampus mainly. Oxidative stress by hydrogen peroxide affects mainly the mitochondria in the non-acclimated hippocampus. Does this mean divers will be less vulnerable to CNS oxygen toxicity at the end of the summer? Well, we don't have enough cases to answer this question, but it is an interesting question. Luckily, we don't have enough cases. So we move on to the protocol of preconditioning. In this case, we will present data that negate the data of the lab of J.D. The protocol of the preconditioning was the same. We exposed the rats to 1 hour at 202 kilopascal each other day for 3 sessions. And then in the 6 days, they were exposed to 608 kilopascal, and the time to CNS oxygen toxicity was measured. We carried on baseline measurements, and samples were taken from the hippocampus and again from the frontal cortex to determine the activity of the enzymes catalyzed, G6PD, glutathione transferase, glutathione reductase, and glutathione peroxidase, all known as ROS scavengers. 24 hours after preconditioning, each rat was exposed to hyperbaric oxygen, as I showed you before, at 608 kilopascal, and the time to CNS oxygen toxicity was measured. Samples were taken again from the same organs, same zones in the brain, and the activity of the same enzymes as in the baseline was determined after the HBO exposure. For the results, the latency versus the two groups, the main two groups, you can see a significant increase in the time of the preconditioned rat from about 12 minutes to something like 21 minutes. And associated with this, we found changes in the enzymatic activity in the hippocampus mainly. This is the enzyme activity. The glutathione peroxidase in the control rats and in the preconditioned rat, it increased significantly. And in the two, surprising for us, the two other enzymes, G6PD and glutathione reductase, was decreased. As we were surprised, we had to give some explanation for this surprise. And just to remind you again, probably the balance between the activity of these two enzymes and the scavenging of the H2O2 is here the issue. And we found a lot of evidence in the literature supporting that reducing the activity of G6PD is a good thing for the patient. It's good for heart attack. It's good for any other injuries in organs. And here too, we found it also in knockout mice. Knockout for G6PD, that the time for oxygen toxicity was longer in this knockout mice compared with the wild type. Why is that? Technology. Okay. As for the nitrosylation of proteins, we found 6 different proteins that were associated with or bound to nitrotyrosine, and the same pattern in the hippocampus, the same pattern was found. Here the pixel per square millimeter ratio in the sham group, and in the control 1 group, which was only exposed to 608 kilopascal. The preconditioned group, you can see a dramatic decline in the rate of nitrosylation, and the control 2 group, which was the group that was exposed only for the preconditioning and not to the 608 kilopascal. In the cortex, we found a somewhat different picture. We found an increase of glutathione transferase, and again with glutathione peroxidase, but we didn't find any change in the glutathione reductase and G6PD. So we didn't get too deep to this picture. And again, the example of nitrotyrosine levels in the frontal cortex, 2 different proteins, 2 different pictures, and we found 8 proteins that were associated with the nitrotyrosine, and the picture was always different. So to summarize the preconditioning investigation, under well-defined conditions, repeated exposure to HBO may have preconditioning effect, providing protection against CNS-oxygen toxicity. The protective mechanism involves alteration in the enzymatic activity of reactive oxygen species scavengers induced by HBO exposure, mainly in the hippocampus. This alteration results in lower levels of destructive RNS and probably ROS, and prolonged latency. And does it mean that repeated dive on pure oxygen protect divers against CNS oxygen toxicity? Well, Jadin will tell you, don't dare to do it. We yet don't know. Again, it's too early to say, but it will be very interesting to follow these 2 opposed findings in 2 labs. In conclusion, these findings are in agreement with the hypothesis that increased production of ROS and RNS is a major factor in the development of CNS oxygen toxicity. It is unclear yet how Hitchcock proteins 72 provide protection against CNS oxygen toxicity, and if it is involved in the protective mechanism induced by HBO preconditioning, something that we didn't measure yet. Generally, the HSP70 and the HSP90 promotes the triage by ubiquitination of damaged proteins, such as those we related to previously. And more specifically, the system of NO generation is involved in the activation of HSP70 synthesis. the association between NO and HSP72 or between nitrogen and the copper zinc sod may be good reason to proceed to the next stage of this investigation. And I want to thank many people that were involved in these two investigations in our lab, Richard Lincoln and Shiran Yechezkel, Frida Neeman, Marina Truman, and Liron Batit. In the Technion to the lab of Professor Yechezkel Kashi, and in Tel Aviv to Professor Ayala Hochman. And thank you for listening.